Welcome back to The Explainer. Today, we're diving straight into the microscopic machinery that literally builds our bodies by perfectly assembling tiny biological Lego bricks. Yep, we're talking about proteins. And look, we know these aren't just macronutrients on the back of a protein bar wrapper. They are the fundamental polymers dictating the shape, function, and very survival of our cells. I mean, from the enzymes digesting your breakfast to the keratin in your hair, proteins are the ultimate cellular multitaskers. Let's get right into it.
Here is our roadmap for today. 1. Amino acids and peptide bonds. 2. The 4 levels of protein structure. 3. Globular versus fibrous proteins. 4. Hemoglobin. And finally, 5. Collagen.
Kicking things off with Section 1, Amino acids and peptide bonds.
So let's start right at the very beginning. Every single amino acid out there shares a pretty fascinating basic structure. Picture a central carbon atom. Now, flank that on one side with a basic amine group. That's your nitrogen and hydrogen. And on the completely opposite side, you've got an acidic carboxylic acid group. Literally every single amino acid in your body rocks this exact same backbone. But here's the catch. The secret to all that wild protein diversity? It's all in the R group. Sticking right off that central carbon is variable side chain. And since we have 20 different amino acids, well, we've got 20 different R groups. It's this one little attachment that completely dictates the amino acid's personality. It decides, whether it acts acidic or basic, or whether it's polar and loves water, or nonpolar and totally repels it.
Okay, so how do these individual bricks actually snap together? Well, when two amino acids meet up, they go through what we call a condensation reaction. Basically, a hydroxyl group gets dropped from the carboxylic side of one, and a hydrogen atom is lost from the amine side of the other. Put those together and boom, H2O, water, is released. What's left behind is a super strong covalent peptide bond linking the carbon and nitrogen. That right there is a dipeptide. String a whole bunch of those together and you've got yourself a polypeptide chain.
Moving right along to Section 2, the 4 levels of protein structure.
Now that we've got our chain of amino acids assembled, well, a linear chain honestly doesn't do much on its own. It's got to fold to actually become functional. And this miraculous folding process happens progressively across 4 distinct structural levels, getting more and more complex. We're going to trace this whole path, from the primary blueprint all the way up to those massive quaternary superstructures. Starting with level 1, the primary structure. This is really just the specific linear sequence of those amino acids bonded together by those covalent peptide bonds we just went over. And the blueprint for this? It's exactly as instructed by your cellular DNA. Precision is absolutely everything here, guys. This linear order ultimately decides every single fold that's going to happen down the line. Next up is the secondary structure. This is where things get slightly 3D. The weak, negatively charged oxygen and nitrogen atoms along the protein's backbone start interacting with weak, positively charged hydrogen atoms. These weak hydrogen bonds basically pull that linear chain into shapes, locking them into tight, spiraling alpha helices or folding them back and forth into flat beta-pleated sheets. And notice, at this stage, those variable R groups, they aren't even involved in the bonding yet. But then, the molecule just explodes into a fully 3D tertiary structure. This highly intricate folding happens because those variable R groups we talked about finally start interacting with each other. Depending on what R groups you have from that primary sequence, you get this crazy mix of weak hydrogen bonds, super strong covalent disulfide bridges between cysteine amino acids, ionic bonds between charged groups, and of course, hydrophobic interactions, where the nonpolar R groups bunch up together inside just to hide from the water. Finally, we reach the quaternary structure. Now, not all proteins get here, but the larger ones do. This is where 2 or more individual fully folded polypeptide chains actually interlock. We call these individual chains subunits, and when they bind together, they essentially form a single massive macromolecule that's ready to tackle the heavy-duty tasks in your body.
So the really crucial takeaway here is understanding the molecular glue that's holding each stage together. Look at the primary sequence. It's just your standard peptide bonds. Secondary structure, that adds hydrogen bonds, but strictly along the backbone. But by the time you hit that tertiary level, the whole squad of R group interactions kicks in. You've got hydrogen bonds, those strong disulfide bridges, ionic bonds that can totally be messed up by pH changes, and hydrophobic interactions. The sheer complexity just scales up so brilliantly at every single level.
Which brings us to Section 3, globular versus fibrous proteins.
Once they're totally folded, proteins generally fall into 2 massive functional categories, and it is based entirely on their final 3D shape. The physical difference is honestly night and day. On one hand, you have these compact, highly folded, roughly spherical proteins. Those are your globular proteins. On the other hand, you've got these long, intertwined strands that look almost like heavy-duty ropes or cables. Those are your fibrous proteins. And as you'd guess, their differing shapes perfectly dictate what they actually do inside you.
A great way to lock in these crucial differences, whether it's for an exam or just to keep it straight in your head, is the SAFES mnemonic that shape, amino acid sequence, function, examples, and solubility. Globular proteins, they have an irregular amino acid sequence which causes them to fold into functional spheres that are generally water-soluble. Think insulin or your enzymes. Fibrous proteins, on the flip side, have a highly repetitive sequence. That makes them structural, like building materials, and because of their exposed hydrophobic R groups, they are completely insoluble in water. No way they're dissolving.
But see this in action with sections 4 and 5, hemoglobin and collagen case studies.
To really grasp these macro-level categories, let's look at two biological heavyweights operating in your body right now. First up, hemoglobin. This is the ultimate globular example. It's a water-soluble oxygen transporter cruising around in your red blood cells. Structurally, it's got a quaternary setup made of 4 polypeptide subunits. But what makes it truly special is that it's a conjugated protein. It relies on a specialized, non-protein prosthetic group called a heme group. That heme group holds an iron 2 ion that actively grabs onto oxygen. And get this, as each oxygen molecule binds, the entire protein shifts its quaternary structure slightly, which actually makes it even easier for the next oxygen molecule to attach. How cool is that?
Now, on the complete opposite end of the spectrum, we have collagen. This is your fibrous structural powerhouse. It forms a really tight triple helix known as tropocollagen, and this super tight wrap is only possible because its primary structure is incredibly repetitive. Literally, almost every third amino acid is glycine. Since glycine is the absolute smallest amino acid, it fits perfectly on the inside of the helix, letting those strands pack together unbelievably closely. Held together by massive amounts of hydrogen bonds, this structure is exactly what gives your tendons, ligaments, and skin their immense tensile strength.
So if you pit them head-to-head, just remember the NOT-MAPS acronym to keep them straight. Hemoglobin, 4 chains, spherical, globular, highly functional, has variable amino acids, relies on a prosthetic heme group, and is totally soluble. Collagen, 3 chains forming a triple helix, long and thin, fibrous, strictly structural, heavily repetitive amino acids, 0 prosthetic group, and highly insoluble. It really is a perfect masterclass in biology, creating vastly different tools for vastly different jobs. Considering how flawlessly these billions of tiny Lego bricks have to align just to keep us alive, I want to leave you with a fascinating butterfly effect to ponder. What kinds of devastating systemic changes happen to the entire body when just a single amino acid in that original primary sequence is swapped out? Well, in hemoglobin, if just one single amino acid changes, the entire protein behaves differently. It clumps together, causing red blood cells to physically deform, and that is the exact cause of sickle cell anemia. One single biological brick out of place, fundamentally changing the entire organism. It's pretty wild to think about. We'll leave it there for today. Thanks for tuning into The Explainer, and I'll catch you in the next one.